from the view of ApoScience, we would like to give a presentation on the new drug development. Just for history, ApoScience is a spin-off company which was originating from the medical university. That's the AKH. And myself, I'm working at the thoracic surgery department, where I had lots of liberty in the last 10 years, where we did all our experiments, we performed our groundbreaking work, as I would say, and then thereafter I had the privilege to develop this entire endeavor in the last 10 years. So basically it's a spin-off at the university, and we're working at the university, and what was very important to me, it's a very strong collaboration between the basic science, the applied science, and also what is also then important to do such a project is to get the money from outside. That means stakeholder money. So this is the appearance of our site of ApoScience. So from my colleague, Herrn Kronberger, Gazalet, Michael Milner, and Helmut Hofbauer, a warm welcome. Of course, we have also Mr. Gugerell and Ms. Lagner, who are also employees of our company. So we have about 11 employees. Four of them are PhD students or postdocs. Also, I want to welcome FGK. FGK is a company which we have chosen to do our monitoring of our clinical study, and especially Mr. Wilm and Mr. Wölfer, thank you for coming to Vienna. So this is actually a slide which is for Franz Trautinger. As I perceive, you have heard and seen a lot of stem cell stories published and also in the media, but I think the first cell-based therapy trial was done by a colleague of Mr. Trautinger, Professor Trautinger, and that is Mr. Holzinger. Mr. Holzinger, and as usual in history and also in science, you always have to go to the origin. And when I was about 27, 28 years old, I found one paper which was published by Professor Holzinger, who is the head of the cardiac surgery department in St. Pölten. And Christoph Holzinger did a seminal work, and I think this is the first cellular-based therapy worldwide published, and that was already in 1994. And just to let you know what he did, he just had patients where he was, as a personal view, I think, suffering because there are no treatment options for chronic wounds. And what Christoph Holzinger did, he applied on chronic wounds peripheral blood mononuclear cells. He stimulated them, and then patients came 2 times a week, and he applied those cell suspensions on the wound. And he was seeing that after 4 weeks, his patient cohorts, which had PFRK or venous ulcer, healed completely. And that is here seen that basically if you apply 2 times weekly mononuclear cells which were stimulated, these wounds were healing completely. And these were the control patients. So these patients had here, this control group was here PFRK, so they called it very like arterial obstructive disease. That's why they call it here some like strange acronym. And these were these post-thrombotic ulcer. And here you can see that this therapy was effective. So when I saw this, it always stayed in my mind. There was, of course, lots of intellectual development between this and that what I'm showing you now, but that was basically the foundation of our patent, of our clinical startup company, of everything what we were doing. So reference to, and this is also very important to me, to those who do these clinical trials. Why I got to know this paper, because I was working with Professor Wollner at the cardiac surgery department, and so this basically the legacy is, for me in this department, it's basically this work. So let's continue. If ApoSec works, this is the hint why ApoSec should be working. That means they applied activated white blood cell suspensions on wounds, and these wounds were completely healing within 4 weeks. and that was with both groups together. Let's continue. So it's pretty much the same story, and I'm sometimes already a bit tired always telling the same story, but in 2001 there were huge papers basically claiming that if you apply mononuclear stem cells in myocardial infarction, you're going to heal infarct. And that really ignited an entire a plethora of publications where basically the entire field was moving in one direction. Take stem cells, put it into every disease you can find. So it could be ALS, it could be myocardial infarction, it could be spinal cord injury, and then you're going to have an effect. And when I was looking at these papers, I got a bit jealous because it were nature papers, and I thought, well, there's one control experiment missing. If you claim that a stem cell is doing the job, then actually you should do a control experiment, and that would be a peripheral white blood cell. And then I had another paper in my mind, which was published by Fadok Hansen, and they basically published also a very nice paper that apoptotic white blood cells are immune suppressive. So then I combined basically these two features, white blood cells plus irradiation and apoptosis, and then I applied these apoptotic PBMC suspensions in myocardial infarction. And that was published then, and that was the result. That was published in 2009, and this was already the time when I was closely collaborating with my colleague Dr. Mildner. We published this in 2009. This is a saline control, and this is apoptotic white blood cells, and you had no infarct scar. And then we started to think, well, this has nothing to do with the cells. It most probably has to do something with the paracrine factors. And this is basically the origin of our patent, and we patented this technology, this platform technology, in 2008. Take-home message, AMI was cured by PBMC suspensions, and that was basically completely against all that what was published at that time. So the hypothesis at that time was then, okay, we take the factors after 24 hours of cultures of PBMCs, we discard these cells, so we have just a biological. Derogatively speaking, people were saying, well, this is a soup. Well, it's a soup. And this you can lyophilize, and then you have got a medical grade compound, which is a biological. And everything what you're going to hear today is about this biological, which was cultured, which is a derivative of cultured white blood cells. So it's basically sort of like a herb therapy. But let's continue. The preclinical research of APOSEC then continued from 2007 to 2015. Here we were able to show that in multiple in vivo and preclinical animal work, it works if you apply APOSEC, which is an acronym for apoptotic secretome. You can basically abrogate damage with myocardial infarction. You can this was a publication by Konrad Hötzenegger, brother of Wolfram, who is sitting here. He showed that basically you can cure myocarditis, experimental myocarditis. Michael showed that wound healing can be attenuated. Not wound healing can be augmented, let's put it this way, but you can attenuate also stroke, which shown by Altmann, and spinal cord injury by Haider. So you've got a multiplicity of preclinical evidence that this drug compound works. And why does it work? Because it has so many features. So we have found in experimentally performed work in multiple labs, because I always think the most dangerous research is always if it's just performed in one lab. So in multiple labs we were able to show that platelet aggregation can be inhibited, neoangiogenesis is caused, and you've got also antimicrobial peptides, which is important for diabetic foot ulcer. You've got cytoprotection, you've got, so we have got, I think we identified about 30 mechanisms. Why does it function in such a pleiotrope way? It is because the content of APOSEC, as far as we know now, are exosomes and proteins and lipids. So many compartments which are produced by our own body are in the APOSEC formulation. This is now the state of the art. We still, Michael and me, we still have another idea what else is in there, but this is not now proven. So this is, and then we had this idea. So, or the vision. What is the first market we can go in? And that's diabetic foot ulcer. That means diabetic foot ulcer is a straightforward clinical trial. You can do a double-blind study, which doesn't cost that much. What can be done by a startup company. However, and this was now for me the greatest challenge. In May 2012, so a lot of years ago, we then invited Mr. Bravery to our company, and he wrote about 30-page sum up, and he said basically, yes, you can produce it. The EMA, the European Medicinal Association, will accept, but you have to follow a lot of steps, and that I'm going to talk to you about. But that was basically a complete new field for me. Complete new field. This is something where I was not aware of. I'm a surgeon, but this was not a completely new dimension. So, what were the features? And that I call a science revelation. If we work in basic science, it's always like, okay, you find out the causation of effect, and that's basically the preclinical research. It's investigator-driven, liberty of science, and academic funding. That means you're working on one side, and you're completely off, and the only person or the only conglomerate of people you have to deal with are basically your peers. The peers who sit with you together in the evening when you're drinking wine, and they're going to say, well, this is a nice research, and then most probably you get it published. But that what really became then for me the new dimension is the product science. It's a completely new word. Nobody knows. I just created it I think about 1 year ago. This is product science, which is regulatory driven, GLP, GMP requirements, safety, stability, quality, and assurance. And that's something medical doctors are not aware at all. And this is sort of like the timeline. So if you go ahead and if you want to end up with a clinical trial, you have to actually go for this, otherwise you will never end up with a story. Just to be a bit polemical, if you talk to politicians, it's always like you have a product, oh, basic science is good, then you go first in man. But this is here never, there's no funding in Austria, there's nothing available now at the moment, so basically it's a dead end. So basically my view is that basic science is a dead end if you don't include this sort of research. And you have to respect this sort of research, and that's what we did the last 3 years. So now I'm coming to the next step. So that Bravery report, the first thing he told us was get a GMP facility. So you have to produce, this is International Commission of Harmonization. It has to be compliant to these guidelines. And that was then the next thing. We had to find a GMP production site to produce our biological. And that was for me already very, very difficult because I did not know that, here we've got a representative, Anja Peterbauer, in the room. We did not know that basically we have a transfusion unit with a perfect GMP facility. And that is headed by, was at the time headed by Primarius Gabriel. Now it's headed by Herrn Magister Watzinger and Süssner and Jüngling. And they have a GMP facility where we can produce our compound. Just to get you through, you've got you need blood. You need blood, and blood. The PBMC are currently a waste product. So you need in the GMP facility you need the PBMC separation. Then you irradiate with 6 degrees, and after that you culture it for 24 hours. Then you have to discard the cells, and that is most important. You have to prove a viral clearance. So there are 2 steps of viral clearance. Just for the doctors here in the room, like immune globulins, they have to undergo a double viral clearance. So we have got also double viral clearance, and we have proven that this is effectful because safety first. Regulatory body wants to have safety first. So just to sum up, ApoSec is a biological produced by living white blood cells, and you can do a double viral clearance. All ATMPs currently at the market don't have viral clearance, so I think there's a safety issue. And what's the beauty of this entire thing is that white blood cells are a waste product. They're discarded worldwide at the transfusion units. So, and this is now Anja. In 2014, in April, Primarius Gabriel, who communicated with Mr. Krasnik, the head of the AGES. He then basically approved this after hearing extremely complex work. You have to have a GMP facility which has to be compliant with the regulations. You have to have a protocol. Then you get, if this is visited by the AGES, they give you a certificate that you're allowed to produce it. But you also need, and that's most important, you need somebody who believes in the project, and that's Anja. So Anja is working with me already like for 4 or 5 years. After maternity leave, she came back, and then now she's basically the mother of APOSEC. I'm gonna refer to another person who's also very important, but that comes a bit later. So second step. We got in 2014, we got the approval that APOSEC can be produced under a GMP facility, and then I did something which is not very well perceived by professors who are working in ethics committees, but there's the possibility to try a new therapy with informed consent. So I knew we've got a double viral clearance APOSEC, and then we applied it on 10 patients, so max 10 patients. After approval, we used our drug compound together with alginate in diabetic foot ulcers, and then we had these results. And that was really astounding. You see here that these are Wagner 10, so very end stage diabetic foot ulcer patients, and they received our drug compound for 1 to 2 times a week, and after 1 month, and we really saw that you had here a massive decrease of massive wound closure. You had new angiogenesis, also new epidermis, and here that was our star. He had a crater, and he was 69 diabetic foot, and then within 1 week this wound closed. So that was performed. We did a photo document on every patient, like every visit was quite a time, but basically we were able to show that this was working. And that was also an argument for me and also for those who were working with me to find an investor. That was the next milestone. Here you see all the important people who were basically doing the job. I just want to introduce them. This is my colleague, Mr. Kronberger. So we both are heading this company. Then we had here Ms. Peterbauer doing the GMP facility, and these three, Mr. Ostler, Ms. Bachmann, and Mr. Sorgenfrei were helping us from the company of Regenold to do the scientific advice, prepare the scientific advice. So we had a 300-page document, and that was and we went then to the AGES and also to the Paul-Ehrlich Institute. Those members are also in the EMA very, very important, and we presented our project to those 2 very important regulatory affairs, and they approved everything what we were showing or what we are showing to you now. Because, and this brings me back to the product science, these 2 agencies allow, based on the ICH criteria and everything what is accommodating this, they are approving basically the trial. So, and they approved our development plan. And I really want to thank here also Ms. Guia here, because she did all the clinical workup. A bit here in the far right. Michael, actually she should be here, but okay. Good. What does it preclude? So what did we? So this I've told you. APOSEC is a biological. It's produced by living cells. It's cell-free, so it's a biological. And you have a double viral clearance. But then, this is the program which we had to follow. Then it's a bit not that well, but observable. But basically, we had to work up. We had to do our 8 toxicology experiments. We had to do viral clearance safety studies where we proved that the viral clearance is effective, and so on and so forth. Ethics committee for phase 1, phase 2 trial. So we have completely done this. But, and this is very important for those who are sitting here as clinicians, this was approved by AGES and PEI. And this document, summing up all these developments, is now being with our company FKK, with our friends from FKK. We're gonna put this into AGES as a first institution regulatory affair, and we have finished now our IMPD document. So this was followed, approved, and now and then we have to wait for about 40 days, and then if the AGES says everything fine, then we are clear, and then Primarius Schwarzenegger and Oberstdrescher can start with their study. So what else? Just to give you some idea why it took that long, because we wanted already to start about 1 year ago, but that was a bit naive. We really had to finish up all this here, which is, I don't want to read it down because everybody can read it by itself. So it just means that this is everything what we had to do. The most interesting thing was the vendor audits and technical agreements, where this becomes like thousands of pages of work. So, coming nearly to the end, Apposcience was always a research company, but then because we're doing now our clinical trial, we have got now a GMP contract manufacturer, which is Ms. Peter Bauer, Watzinger, Linz, and Apposcience had to become a pharmaceutical company. So that means this was the legal requirements, and we are now basically a paragraph 63 company, where we are legally seen as a drug producing company. And this again, I reiterate here, we had to also employ a qualified person. A qualified person is basically the person who gives the batch release of our APOSEC drug compound. And this is now the person who was basically standing on the pivot, because he's the guy who basically approves that everything is produced according to GMP, and that's Mr. Seibold. So we have got this construct which is approved by Mr. Karsnik. Markus Seibold is working as a head of quality control. He's a sub-quality director in Linz. So he's employed in Linz, and he's also our qualified person who releases our drug compound for the company. That's why the reason why we are a bit delayed, because we wanted to start in August or something like that. We had AGES inspection in December, but when the AGES inspectors came to Anja and her team, they told them we're gonna audit just their common product, but with APOSEC they're gonna come another time. So that was in April, so that was a 6 month where basically everything was a bit postponed, but we have got good indication that the production of our compound will start in August this year, and the release of the first batch will be done by Markus by December. This is the clinical trial. Professor Dozent Guya is going to talk about this, but basically we're going to have a lead-in phase in Linz and in Klagenfurt, and then we have got Primarius Trautinger. He's the head of the DSMB, that's the Data Safety Monitoring Board. After 12 patients, then the data will be observed, no adverse events, and then hopefully we have no indication that there will be something else, and we have got already some efficacy data. So basically we have got 12 patients here, and these 12 patients, 9 are getting the drug compound, 3 are control. And then we're going to enroll the entire trial. These are just the data. Everybody here in the room will going to be supported and will get these data. These are just here, mid of December, possible first patient in, but realistic is birthday of mine. That will be here. It's one orthographic mistake, but okay. So here now we have the first safety first. That's these famous word of Ilona Reischl, AGES auditor, good manufacturing practice. So we had here the clinical trial application, investigator brochure, IMBD in June will be handed in by FGK this week. Here in August till November, we're going to have the placebo plus APOSEC, the release in December, and first patient in, I think, most probably in January next year. 15 years of development. It's a PPP investment, so public-private partnership investment, and that's really the way of a bench to clinical trial. What Alfred and me had this idea just 2 days ago, from start of the production, we're going to have a newsletter, just a PDF sent to our partners, where we're going to say this is the status, and then I think from August onwards we quarterly invite all our colleagues to get patients for our clinical trial. However, highest command is safety to each patient and top quality, and that's why it took that long. So last slide, a reference to those who made this possible. You can see Folab Medical University gave the opportunity, Christian Doppler Laboratory, FFG, basic program, Wiener Wirtschaftsagentur, ApoScience ongoing, and then also Kepler University and Elisabeth Thiene in Klagenfurt. And I think this is the end. Thank you.